Keller, before we start, I just want to say 38 participants is the highest we've had this semester.
Oh,
Yeah, this is so before Dr. Parks even does her introduction, we can already tell your talk's highly anticipated. So
That's wonderful.
right. I hope it's interesting. Thank you very much.
No pressure. Welcome, everybody. It is my pleasure to introduce Dr. Kathleen Keller. She received her PhD in nutritional sciences from Rutgers University in 2002. And I think that was under the mentorship of Beverly Tepper. Is that right?
Yeah.
A very well-known food scientist. And then Dr. Keller completed her postdoctoral training at New York Nutrition Obesity Research Center. And there it looks like you were working with Miles Faith.
Yeah. And then Harry Kiselev. Miles left about a year into my postdoc. So it was sort of a joint effort.
Yeah. This really focused on obesity in children. And from the start of her training, she's worked at this intersection between food science and nutrition. She's currently an associate professor at Penn State with appointments in both nutritional sciences and food science. And she's also affiliated with the Social Science Research Institute. She directs their Metabolic Kitchen and the Children's Eating Behavior Lab, a shared research facility dedicated to the study of eating behavior and nutrition in children and families. Her primary research questions involve characterizing individual differences that influence the development of childhood eating behaviors and susceptibility to obesity. And her current NIH R01 is using neural imaging to understand cognitive behavioral factors and environmental factors underlying the development of eating behaviors. So please unmute. I'm an old school and I would like you to unmute and welcome her, please, with some clapping. Can we hear it? We're happy that you're here. Thank you so much.
Thank you. Well, it's a pleasure to be here. Wish I could actually be there, but it's a pleasure to see all of you. So I am going to share my slides with you, but I'm happy to discuss anything if you just want to interrupt me, if there's any questions that you have at any point or anything you want to learn a little bit more about. So what I'm going to do today is kind of give you an overview of the work that we do and, some of our projects that we've completed. And at the end, some of the work that we're doing now kind of to move this to the future. So just to give you an overview, I'll talk a little bit about kind of the big picture of our research, why we're interested in studying children's individual responses to food cues. And by food cues, I'll be introducing sort of these cues in our obesogenic environment that we see and experience quite a lot, and that we think that some children are more drawn to than others. And so I'll give an overview of some of the neural differences that we've found in response to food portion size and food marketing. And then if we have time at the end, I'll talk a little bit about the cohort studies that we're doing now, the NIH-funded cohort studies. So this is just a picture of our feeding laboratory. We built this when I came to Penn State. So we have a waiting room for the families and a couple different observation rooms. We do a lot of video coding of behaviors. So we have hidden cameras where we can record what the kids say and what the kids eat. And then our kitchen here. And we have a pretty big research group now. This is actually a couple years old, but our lab coordinator, Terry, manages the kitchen. And then Kara Hickok is an undergraduate. She was an undergraduate at Penn State. And now she's running our food and brain study, our longitudinal cohort study. Elena Pierce is a postdoc from the area of cognitive neuroscience. And then I have 3 doctoral students and another one joining next year, a master's student, Wei Lin, who I've actually never met because of the pandemic. So she's in China and she'll be joining us over the summer. And then we get a lot of undergraduate students who are from many different departments. So one of my favorite things about what we do is it's very interdisciplinary. So we have students from psychology and nutrition and food science and human development and family studies and sort of on and on, who are interested in the work that we're doing, which is great. So our overall question, sort of the theme of our lab, is understanding how eating behaviors develop. And when I was trained with Beverly Tepper, we were looking at genetic taste factors that influence what we like, what children like, what foods they don't like. But since coming to Penn State, in large part because of the position that I was on, and then really becoming interested in the brain, I've incorporated a lot of neuroimaging in our research. We also look at factors in the food environment. And so ones that we've been most interested in are food advertising, portion size, parental factors, and issues related to food accessibility. And then we also look at individual differences in temperament and traits and cognitive development. And so all of those sort of individual differences, the hope is that we can characterize phenotypes of children who are at risk for overeating in the current food environment. And that because of that, we can potentially identify modifiable traits that we can intervene on. So I won't be presenting any intervention work. That's probably a more limited part of what we're doing now, but that is the ultimate goal. So some of the concerns we have in terms of the obesogenic environment, for me, have to do with the constant surrounding of food cues. And by cues, I'm talking about exposure to advertising, pictures, smells, sight, sound of food that can be very tempting for some people. And now we also have added on to that social media, Instagram, cooking shows and cooking channels where we can be surrounded by food and food images really 100% of the time. And so children who are very vulnerable to overeating in response to these cues are ones that are the most vulnerable to gaining weight in the current environment. So I'm going to just start off to get you thinking about how you might respond to food cues. I'm going to have you think about, rate your current hunger. You can do this in your head. So on a scale of 1 to 10, with 1 being least hungry and 10 being most hungry, how hungry are you? On a scale of 1 to 10, how much do you like pizza? And how much do you want pizza right now? Okay, so think about those things, and then I'm going to show you a quick video. And you all see this commercial?
Yep.
Okay.
Woo! Got some fresh new hotness coming in. Detroit style pizza. I'm talking crispy, cheesy edges. 78, 79, 80 pepperonis. Little cupped ones, too. And no, it's not a mistake. The sauce goes on last to keep the crust light and airy. This might be the best thing Pizza Hut's ever made. Can the Hut out pizza itself? Whoa. Try the new Detroit style today. No one out pizzas the Hut.
I can see where you're going with this.
So, it's a little bit before dinner. It's almost dinner time for me. The assumption has been that, that adults are able to have the cognitive resources to resist food and food cues. Somebody said, is 10 the maximum? So, and Pizza Hut's not particularly my favorite place to eat pizza, but obviously when they give you that shot of the cheese kind of dripping away from the crust, it's very tantalizing, and it can make someone who's even resistant to those types of cues drawn to them. And so, as a mother of a young child and then someone who does research with children, I'm very concerned about the amount of exposure that kids get to food and food cues in our current environment. So let's go back to the presentation now that I've kind of got you thinking about these things. So hopefully, just by doing that quick experiment, you saw that the extent to which we like foods and how much we actually want them can change. It's not a constant. Even though we might not be thinking of a food, if we see an advertisement or a picture, all of a sudden we start to get hungry, and maybe we go to the store, get the motivation to make an order to actually to get a food just based on seeing that cue. So this is one of the reasons why we use methods of neuroimaging to really get at these differences in susceptibility that people have to food cues. And this is really based on the assumption that we learn or are conditioned to respond to food cues. And we can think back to Pavlov here, where you can train a dog to salivate when they hear a bell based on, pairing the bell with the presentation of food over time. And animals would do the same thing, of course, with a cue that might be paired with alcohol. They will eventually place a lot of effort to find that cue, whether or not it's followed with alcohol or not. And these same sorts of cues are present in our own lives. And so, of course, marketing and branding is a big one. But we also have other types of cues. So social norms, for example, or just visibility of food in our environment, special kinds of dinnerware, or package and portion sizes, perceived variety or palatability or labeling of foods. So you can think of many other types of examples that across the meal, whether we're talking about initiating a meal or planning or consuming or the ultimate end of the eating episode, we have a lot of different cues that can impact our choices during this time. And they can easily sort of override our internal ability to counteract those cues. So in terms of exposure to food cues, we know that just being exposed to different pictures of foods or advertisements can increase salivation and heart rate, and can increase our gastric activity as we think about digesting or metabolizing those foods. And it also can increase neural activity and dopamine centers of the brain. So dopamine is released. And of course, this is a reward-related chemical. And so these types of experiences can really increase the desire to go and seek food. And so the assumptions that we have when we do neuroimaging research are really to get at these conditioned experiences that we have formed with food cues and to try to understand differences in susceptibility to those. And of course, we have a lot of flexibility in the types of neuroimaging work that we can do now. It used to be that you had to wait for someone to actually get a tumor or a lesion through their brain in order to understand the role of the brain in eating behavior. And a great example of this I like to talk about is the Gourmand syndrome. So tumors that are, in some cases, impinge the amygdala and the temporal pole can result in drastic changes to the personality, where someone will all of a sudden become obsessed with food and with eating. And in some cases, there are cases of people becoming food critics because they are so attentive to the various aspects of food and so interested in it. And as soon as you remove that tumor, these personality traits go away. And so we can see how different areas of the brain are involved with very unique and sort of unusual characteristics and fascinations with food. Fortunately, we don't have to wait till children develop a tumor or a lesion through their brain, because now we have really nice methods of neuroimaging. And I'll just quickly kind of go through what our main method is, which is blood oxygen level dependent functional magnetic resonance imaging. And I apologize if this is, if you know what that is, this is, but I'll just give a quick overview of what the method does. In food cue reactivity research, we often show pictures. We sometimes give different tastes of chocolate milkshake, for example. And with that, there is some neuronal activity that takes place. And that is coupled with an influx of blood and an influx of glucose. And that results in blood flow that rushes to the area of the brain where there's neuronal activity taking place. And so this increase in blood flow results in a exchange, an increase in the amount of oxygenated relative to deoxygenated blood that results in this 15-second peak and what we call the hemodynamic response here. And it's this hemodynamic response that can be detected by the magnet in the MRI. So when you're looking at an MRI result of the brain, this isn't actually directly measuring blood flow, but it's measuring a correlate of what we assume to be blood flow. And there have been a number of validation studies to support this. So this is what we infer to be neuronal activity. And so to understand a little bit about how different types of physiological signals or external signals can impact areas of the brain, you can vary the conditions. So you can scan people when they're fasted or fed, or you can scan them after a delivery of a satiation signal or satiation hormone, or you can scan them under situations where they might be stressed, or look at some of the individual cognitive factors or habits that impact their response to these different food cues to infer a little bit about the role of the brain in responding to these cues. So the question, James, will the food cue you use to induce a neuronal and hemodynamic response in bold fMRI be unique to the specific study, or is it analogous across studies? So that's a great question. When I got into this work, I was brand new to neuroimaging, and I didn't have any background in it at all. And I was trying to think about what me, as a nutrition person and a food science person could really bring to the field. And a lot of what we've done is when we're starting the work, a lot of people would just take Google image searches and look for pictures of food online. And so they would be not standardized for portion size or not standardized for branding. In some cases, you'd have food brands and other pictures not. And so we've really spent a lot of time standardizing images and getting a professional photographer to take them so that we have sets of images that are consistent and reproducible. And so now if you go online, you can actually get image sets from investigators that you can use across studies in order to get more reproducible data. So the images that we're getting in MRI are based on, the interpretation is really based on contrast. And so some of the work that I'm going to talk to you about is comparing, for example, the regions of the brain that light up when someone's looking at large portions of food compared to small portions of food. Or we've looked at how kids respond after viewing food commercials compared to after viewing something that's equally rewarding, but is not food. And that might be like toy commercials in the case of children. Or we've also done other protocols where children win in the scanner through gambling paradigms. They win either food or money. So again, they're two reinforcing, rewarding stimuli. And then we compare the differences in brain responses between these conditions. So in order to really, I think, get the most out of imaging, you have to have a clear hypothesis for how you want to, or framework for how you want to understand the questions that you're asking. And so a lot of the hypotheses that we've been asking in our lab are framed around the dual systems or reflexive impulsive model. And this simply suggests that there's a balance between the control centers of your brain coming from the prefrontal cortex and the reward centers of your brain. And it's really not one or the other, but it's when there's an imbalance. So a reduced control response combined with an increased reward response that might make an individual vulnerable to overeating. But that's a very simple way to look at the brain. And so I do want to recognize that the appetitive network of the brain is very well studied, and it is consistent at this point. And so we can think about homeostatic signals coming from the hypothalamus, of course. And then we have reward and drive signals from the striatum, which allows us to not only feel reward, but to activate that reward to action, to actually be rewarded enough by a stimuli, but then to actually have the motivation to pick up the phone and order that pizza. The orbitofrontal cortex and the ventral medial prefrontal cortex that I'll spend some time talking about today are areas of the brain that are involved with the subjective value of food. And so that can change with physiological signals, and it can also change with, of course, the palatability of the food. And then the insula and the amygdala and hippocampus, which are sometimes called the ingestive cortex, but the insula is important as a primary taste cortex, but it's also really important for this ability to interoception, or to perceive external signals accurately. And so if you think about the ability to respond to hunger and satiety cues, a lot of that is going to come down to your interoceptive abilities. And then we have our self-control network, the anterior cingulate cortex and the dorsal lateral prefrontal cortex. So those are sort of the frontal lobe areas of the brain, and are all interacting with these lower sort of limbic areas of the brain. And so there are many regions of the brain that respond to food cues, and I'm going to focus on a few of those today. I'd also add the cerebellum to this picture, because the cerebellum is at the base of the brainstem, but it also has connections to the hypothalamus, so it's involved in satiety signaling, and it also has connections to the self-control related areas of the brain. And it used to be something that we would ignore in neuroimaging research because it would come out on almost every single food cue study. But we're now starting to get very interested in this particular region as a key area for processing of satiety signals. So before I get into talking about the data, I want to just provide some of the thinking that we have when we've been developing our studies. And one of the first areas or sort of obstacles we faced is that we can't actually have people eat a meal when they're getting their brain scanned, at least not if we're using bold fMRI. So how can we get information from their MRI response that is going to be informative when we think about their eating behavior? So we could do that in a couple of different ways. One is to show them some pictures in the scanner, and then do a whole brain analysis where we see just kind of the overall pattern of activation in when they're looking at high calorie foods relative to low calorie foods, for example. And then we can relate those patterns to intake at a meal in the lab to see if there are certain patterns that predict overeating and other patterns that don't. We can also develop specific a priori hypotheses about regions that we think are going to be involved. So again, we can show people different images in the scanner, and then based on our knowledge of the appetitive network, we can select activation in a few regions that we're interested in to see if they explain variability in intake behavior. The reason I point these out is because one of the things that we deal with in MRI is that when you scan the entire brain, you're dealing with 300,000 voxels of information. And so a lot of our early studies that only had 12 or 14 individuals in them were vulnerable to false positives because they tested all of those 300,000 voxels. And when you do that many statistical tests, of course, you find something. And so I'm more of a fan of, well, we've rectified a lot of these issues with newer, larger sample sizes and newer analytic approaches. But I'm also a fan of these smaller scale sort of region hypothesis-driven analyses. So I'll show you some of those today. So I'll talk a little bit about the data that we've collected on some of the studies that I've done so far at Penn State. And whereas my work prior to Penn State was in early childhood, when we came to Penn State, I got interested in studying middle childhood. And part of this was feasibility of scanning, because about age 6 and 7 is about the youngest age we can get really good data from children in the MRI. But there are also a lot of other things going on in middle childhood. And kids are developing more autonomy over their food choices and eating fewer family meals. The brain development is sort of preparing for puberty, where we see sort of a protracted development of the prefrontal cortex and an increased development of the reward centers. There's a lot of media use, and this has gotten increased with COVID because kids are watching more media and overall preparing for puberty. But we're also seeing the start of disordered eating, and in particular, loss of control eating, which is the perceived inability to control or stop how much you're eating at a particular meal. And the other area that I wanted to study when I came to Penn State is the portion size effect. So this very robust phenomenon where if you serve people more food, they eat more. And this is observable in preschool children, school children, and adults. And it's very robust, and it's increased, but particularly with energy dense foods that are highly palatable. And before I came to Penn State, I was really interested in a lot of the work that Barbara Rolls was doing. And so she had not really been looking at the individual differences on the portion size effect. And so we've developed a really nice collaboration now where we're identifying individual level factors that increase or decrease susceptibility. So in the lab, we study the portion size effect by randomizing children or adults, in this case, 7 to 10-year-old children, to meals where they all the foods are increased in portion size. And they don't know that this is actually happening because they get them in a random order. In this particular study, we had 3 different energy dense foods, a cake, garlic bread, and macaroni and cheese, and 3 different low energy dense foods, broccoli, grapes, and cherries. And we give the children an opportunity to eat as much as they want while we read them a story. We also video record their meal and eventually code aspects of the meal microstructure like bite rate, eating speed, and other variables that we think might be relevant for predicting individual differences. So overall, we see what we would expect in this type of environment. Kids tend to eat more when they're served more. But what we also notice here is there's a curvilinear relationship. So as portions get very big, and we don't know really why this is, intake tends to level off. We also see, if we look just at the high ED foods alone, a reliable sort of portion size effect here. And it's less robust with our low energy dense foods. And this is work from Barbara's lab that I collaborated on, but in adults, the effect of portion size and the effect of increasing energy density has sustained effects on overall intake. So this is actually in preschool kids, and this is the consumption, daily consumption when portions were increased by 150% of baseline. And this was the baseline portion consumption across 5 days. The same types of effects can be seen with increases in energy density. So on the low energy density condition, over 5 days, kids' intake is lower, and it's higher on the high energy density conditions. So over time, children don't adjust to these conditions, or at least not over the 5 days that we've observed them, and they continue to eat more than what they eat on the baseline conditions. But one of the interests that we have is that there is substantial individual variability in the response to this. So when you're just looking at the mean responses, it looks as though everyone's susceptible to the portion size effect. But if you can actually look at individual responses, this is across 4 portion sizes, what you see is that there are some children that are very susceptible and others that don't actually seem very susceptible at all. And so what is it about these kids that differentiates them from these children down here? And is there something that we could potentially use in interventions that these kids are doing that could help kids who are very susceptible? So now I'm going to focus on a couple different regions of the brain when I talk about some of the results that we've seen. I'm going to be talking about the ventral medial prefrontal cortex and the orbitofrontal cortex, which are involved with the subjective valuation and overall reward value of food. And the dorsolateral prefrontal cortex, inferior frontal gyrus, which is involved with self-control. And then I'll also talk about the cerebellum, which is involved with craving and connections to these self-control related regions of the brain. And in this experiment, the overall goal was to see whether food cue reactivity, in particular in response to portions of food that were either large or small and varied by high calorie and low calorie, if these responses to these conditions would explain individual variation in our portion size effect here. And so would we be able to see brain-related responses that made some kids more susceptible? So they might, particularly at the lower portion sizes, increase intake very rapidly, and other brain patterns that make some kids less susceptible. One of the characteristics that has a robust influence on the portion size effect is perceived loss of control eating. And these children are 7 to 10 years of age, predominantly healthy weight. And they were in a study where they were randomized to consume meals in the lab. And we took a number of other measures as well. We asked the kids on one of the visits to report whether or not over the last 3 months they had perceived the inability to stop eating or control the amount that they ate. And this is a predictor of the development of binge eating disorder and is thought to develop in middle childhood. And so when we asked the children this, about 15 of them, about 30% said yes, they had experienced loss of control eating, and 34 said that they had not. And the children that experienced loss of control eating in the last 3 months, you can see we're much more susceptible here to the effects of portion size, both for the meal overall and for just the high energy dense foods here. And we also got some clues that the children with loss of control eating also have altered brain processing of portion size. And in particular, we saw in the cerebellum there's no difference among the children who have no loss of control, but among the children who have loss of control eating, they have differences in their processing of these images in the cerebellar region. We don't know if this is causal, but it's an area that we're following up with. And at the beginning of the talk, I had said that the cerebellum has connections to these self-control regions of the brain. So I wanted to do some exploratory analyses to see if we'd see similar patterns of response to large and small portions in the dorsolateral prefrontal cortex. And they're not as robust, but we do see very similar responses with large portions in our loss of control children being showing lower activation in this region than small portions. Now, we've also, this is a study that, or this is a combination of data that we've looked at from all the studies that we've done. So we have about over 130 kids in this set, but we also see structural differences in the brain of children with loss of control eating. And so we see lower volume in the left hippocampus and in the nucleus accumbens and caudate, which is a reward processing center, and overall in the whole brain. And it's interesting that these kids are primarily healthy weight. So this is just based on their sort of early indications that they might be vulnerable to binge eating later on. And there's a question. So did you collect info about the parents of the children with loss of control, such as BMI? So we do have information on the BMI of the parents in those studies. And in our current study that I'll talk about if I have time at the end, we are actually splitting the study by risk status. So we can look at whether or not the parents are high risk for obesity or low risk based on parental weight. In this particular study, we controlled these results for the child weight status, but we didn't control them for the parental weight status. So it's a good thing to look at. So that was sort of a aside to look at kind of overall structural differences, but going back to the brain responses that kind of predict individual differences in the portion size effect, we found that children who have higher activity in the inferior frontal gyrus, which is a control area of the brain, seem to be less responsive to the portion size effect based on their intake responses here, which don't seem to increase very much with increasing portion size. On the other hand, children who had low activation in their inferior frontal gyrus seem to be more susceptible to the portion size effect. We also saw that children who had higher activation in reward and value related areas of the brain, the orbitofrontal cortex and the ventral medial prefrontal cortex, seem to be more susceptible to the portion size effect than children who had lower activity in these regions. And interestingly, in another study that we did where we were looking at children's response to winning food or money in the MRI scanner, we found that children who had greater activation in the orbitofrontal cortex when they won food compared to money also ate more in the laboratory. So these measures predict measures of eating behavior in the laboratory. So where we've been, we've found in our studies that the ventral medial prefrontal cortex and the orbital frontal cortex activation in these areas seems to correlate with increased intake in certain conditions, and activation in the self-control region seems to correlate with decreased intake in certain conditions. And these relationships are independent of child weight status and how much children like the food. So you could say, so what, they correlate with laboratory measures of intake, but does that mean anything with respect to long-term weight loss? This is not data from our lab. This is from Milan Dagger's lab, but this is activation in the cognitive control regions, the dorsolateral prefrontal cortex. And it's predicting the amount of weight loss at 1 month and 3 months. So they seem to be also predictive of longer term weight outcomes. And it's obviously not just activation in the dorsolateral prefrontal cortex, but also the balance between activation in these reward centers. And so in this particular study, and there are a couple others to show this as well, but greater activation in the control centers relative to these value and reward centers was related to lower likelihood of enacting or giving into cravings. So interestingly, we have found that one of the things that lowers activation in the cognitive control regions is food marketing. And so this study is from a separate set of kids, 7 to 10 years of age. They came in on 2 different visits. In one of the visits, they were scanned on both times. And in one of the scans, they saw toy advertisements. And then they were shown pictures of foods. And then on the other visit, they saw food advertisements. And then they were shown pictures of foods. So after seeing food advertisements relative to seeing toys, they had lower overall activation in these cognitive control regions of the brain when they were then subsequently shown pictures of food. And in this particular study, we had a really nice split of children with overweight and children with healthy weight. And in the children with overweight and obesity, what we found is that food commercials, not only did they decrease activation in these cognitive control regions, but they actually increase activation in regions of the brain that are involved with reward and subjective value. So the research that we've collected so far is leading us to this idea that there's a vulnerable neural phenotype that is described as reduced engagement of these control regions in response to food cues and increased engagement of reward and valuation regions of the brain in response to food cues. And from other studies, we know that this is predictive of weight outcomes. And food advertising seems to reduce responding in control areas and increase responding in reward areas. So based on these results, is there anything that we can actually do to intervene? Well, in the future, one of the approaches could be to do neuromodulation, where you can actually train people to respond differently in areas of their brain to see if that can impact their sensitivity to different food cues. I have a couple more slides to show with the R01 that we're doing now, but I can also take questions if you think, if I'm running out of time. Do I have a few more minutes?
Yeah, you do. And if this is hot off the press, it'd be great to see.
Okay. So I'll just talk to you a little bit about our current study. So we call it the Food and Brain Study. We're actually in the last year of funding, but we're quite a bit behind because of COVID. So we have 120 healthy weight kids, 7 to 8 years of age, and the kids are split by risk status. So half the children have parents with obesity, and the other half have parents with healthy weight. And we have 6 baseline visits where the children come in and get a DEXA scan, and they have a number of different eating protocols where we measure their hedonic eating and homeostatic eating. We measure physical activity, and we measure a number of cognitive self-control and other traits that can increase or impact executive functioning. And we also have the children eat meals that vary by portion size. And then after a year, they come back and they get more anthropometric scans and follow-up eating behavior measures to see if these baseline traits predict likelihood of weight gain during this time. In this study, we are focusing a lot of effort part of this was ongoing anyway, but when we were shut down a year ago, we had a lot of undergraduate students who wanted to keep busy. And so we are coding aspects of the meal microstructure. So bites, sips, latency to first bite, meal duration, anything the child says. These will hopefully identify modifiable eating behaviors that can eventually be used for intervention. We spent a lot of time at the beginning of the study developing our own image set and validating this and having children rate it. And so these are just a few pictures, but we, in the MRI scanner, the children see small and large portions of foods that are high energy dense and low energy dense. And they also see office supplies of varying sizes. We also developed some measures of inhibitory control. So this is a stop signal task, but it's a food-based stop signal task. The children have to press a key, and then when they see a cloche come down, they have to inhibit their response. And so this is an indicator of executive functioning and inhibitory control. And what you're seeing here, this is analysis that we've done a while ago. We actually have more data collected now, but higher scores equal worse inhibitory control. And we're not seeing any difference with energy density or portion size, but we are seeing a trend for high risk kids, even though they're healthy weight, to have worse inhibitory control. And we also are looking at working memory, which is another component of executive functioning. And to do this, we use an NBAC task where children have to let us know when they've seen a letter, either one previously or two previously, and the two back. And we're already seeing that the kids who are high risk have poorer working memory, particularly on when the task gets a little harder. So on the one back, everybody gets them right. But as soon as that becomes a little harder, the children who are high risk are showing poor working memory. And so to summarize, and then we can talk about questions, the prefrontal cortex is emerging as a predictive marker of consumption, independent of weight loss, and is predictive of success in weight loss diets. And so neural markers can be integrated with other types of biological markers or cognitive tasks as ways to develop more effective interventions. And that's really the goal of where we're heading. We also need to understand resiliency factors. So we just got a grant funded that will start in May to look at a very similar type of design, but to look at neural and cognitive factors that promote resiliency to food marketing cues. And so we're very excited to start that study. So any questions? I'm happy to discuss any questions you have, or that was kind of a quick overview and a lot of different information in it. But
thank you so much. That was wonderful. There's a couple of questions, and I'm not sure if you covered them or not, but there was a question about the parents of children, and I wondered, even though you answered that, what is known about the bold patterns in families? Is a good portion of this inherited, or is this learned?
I don't really know if there's much that's inherited, because it's very context specific and it changes over time. So developmentally, it is it's sort of specific with development. So I don't know that you would expect to see similar types of bold patterns in parents and children, but it hasn't really been studied that much, honestly. one of the things that we're going to do in our new study, hopefully, is do some metabolomics and microbiome collection of the children. So we're hoping to get a supplement to look at that.
Oh, cool. Kathleen's asking if you experience a lot of movement artifacts because you're working with children. I can't imagine, if they'll sit still and kind of how does that wiggling impact your data collection?
So we spend probably a full visit with them. There's a full visit dedicated to training them in a simulation scanner. So it mimics the sounds. We can play the sounds for them. We can show them what they're going to, similar things to what they're going to see, and ask them questions and tell them how to answer without moving their head. So we also have a program that we haven't used much, but other people have, that can chart movement and train. They get a little beep when they move too much. And so we found that we have about 80 to 90% rate of success based on our training protocol. But if we were to do younger children, or I have a colleague who looks at children with autism, and she has used some of these other more sophisticated methods. So
did that get at your question, Catherine?
Yes. Here too, we have a mock scanner. So you're able to put subject in a mock scanner just to kind of orient them before they have to get into the actual scan.
Yeah.
Yeah.
Do you think there's a question about, kind of thinking about whether loss of control or is nature or nurture? And, we eat as kids what our parents buy or make, right? What is the thinking in the field now? Can we reduce loss of control in a little kid who exhibits this early on?
So there's a couple questions that I think, one is whether or not kids at this age truly understand the concept of loss of control. So that's been debated a little bit. So particularly in girls, there is a lot of, modeling off of the mom. And so if the mom is dieting, girls will tend to start dieting sooner. And so I think there's a fair amount of these behaviors that are being picked up from home. There are also some genetic markers of binge eating, and we don't know exactly how. We haven't tested those yet, but I think it is something that we've been interested in doing.
Do our siblings, another question, are siblings more like each other than two kids from different families?
The work that I've done with siblings in my postdoc was, what we found is that the parents will feed siblings differently depending on their likelihood, the parents' likelihood, whether or not they think they're going to be overweight or not. And so if a parent is more worried about a particular child, they will restrict them more, they'll pressure them more, so a lot of it is really specific to the child. That's one of those areas where even if there are genetic similarities between the siblings, the perception of the parents is very important in the feeding relationship.
Yeah. There's a couple of questions about physical activity. So you put a tracker on a kid and get their spontaneous physical activity. And how does that, is there a relationship between that and the brain areas that light up with food?
I'm not entirely. So I guess what we've looked at is how imposed activity affects intake directly after. And so what we found is that kids who perceive the activity to be harder eat more, but the activity itself doesn't affect intake, but it's all the perception of how hard they worked, so there's a big cognitive element there.
That is really interesting. I guess there's lots of great questions here, and these students have been thinking about energy balance and obesity this semester. I guess, can you kind of put in context for us where this field is? Because these are very intensive, expensive studies and you don't have a sample size, your sample size are really large compared to what has happened in the past in studies.
Yeah. I think it's getting increasingly interdisciplinary. And so just there's more and more of an understanding that just looking at energy balance, just looking at physiology is going to be incomplete. So you have to really look at other types of behaviors, other types of characteristics and traits. So I see that happening. And I think that, there is a need to, we don't really have the long-term studies that we need to really draw the conclusions that I think we want to make. So there's a need to do really, longer term studies to understand how these things actually play out beyond short term in the lab.
Yeah.
And, really everybody, people are really moving towards big data, open data, having data that, because the stuff that we do is so expensive, it really is important for students to learn how to do secondary data analysis, because NIH is really moving towards all of this data being available online, open science. And then it could be years to really look and analyze at all of it. And so having the skills to do these types of secondary data analysis will get us through pandemics, and also, get us through these. Hopefully we won't have to do that soon, but I think it's important.
Pre-pandemic, what's the most challenging part of this work, and what's the funnest part?
I think the most challenging part is I is always feeling like you don't know very much in any one particular field, because there are 8 or 9 different fields, subspecialties that are involved in this. And so always kind of feeling like you are comfortable with never knowing enough of what you need to know, but the big picture pieces to put it together. And that's been, I think, challenging and an adjustment, but I think it's also been one of the things I like the most about it, because you're always learning new things. And, I still, my favorite part is probably just the training students and watching them get interested in their fields of specialty. And they're so much quicker and better at doing these things than I am. So I really like to see that.
Lots of data. How, I can learn to do an enzymatic assay in 2 days. How long do you feel it takes in training to really master these techniques?
It's I don't know that it's possible to master them, because they're always changing. So you really have to be involved 100% in the changes. And so I just essentially just work with good people that are involved in it. I realized early on that I am not going to be the person who's able to do it all myself, but I can work with really good people.
Yeah. Is it a couple year path to really wrap your head around the brain regions and all of that?
I would say at least 3 or 4 years, I would say, to where, but I think it's really the analysis part is complicated.
Yeah.
So.
Yeah.
Jaffna, did you have a question, Jaffna?
Yes, thank you. This is the great presentation. And as you're presenting, I was thinking of that video. I'm sure most of you've seen it. It's about, this video shows American kids looking or watching videos of what, children from other countries eat for breakfast, for example, and stuff that we eat in India and in China. And they were like, ew, like you would never eat that. So how do you control for that cultural heterogeneity in your population and in your analyses? So the kind of foods that you showed in your presentation, those are typical American foods, but a lot of the population, maybe some of your participants don't grow up eating those foods. And do you think that would influence, these bold responses?
Yeah, definitely. That's a great question. We can control for some of these things in a couple different ways. One, in our screening, so we sort of ask, will your child generally kind of eat those foods? Are they on a particular diet, that might really make it so that they're not going to eat or like the foods? We also ask the parents to kind of report how familiar they are with these foods, and we adjust for some of these measures. And we also ask the children to rate like how much they like the foods themselves. We unfortunately have a lack of cultural diversity around Penn State. It's very challenging for us to get a lot of, but even so, we still have differences in family eating patterns and things that we do try to control for. And so those things are extremely important, as you can see in those reactions those kids have in that video. They're hugely important.
Yeah, thank you. And as you were saying, as the field progresses, hopefully we can sample, people from different populations.
It's one of the, it's a huge gap in, not in the knowledge base, and something that needs to be done.
Well, thank you very much. This is a really interesting talk, and appreciate your time today with the students as well. Please, everyone, show her some appreciation for this science, and thank you.
Yeah, thank you so much for asking me. This was a pleasure.
Yeah.
Bye-bye.
Thank you.
Bye.